Okay. So what I wanted to do is talk about how you can learn about nervous systems from watching animal behavior. And I promised Bruno that I would work in something about bloodsuckers. So I said, when it comes to the nervous system, leeches don't make sucky teachers. So the reason for that is that this is a leech, Hirudo verbana. This is the one we work with. And this is a medicinal leech. And they're a sanguivore. That means that they eat blood to survive. Now, the thing that has captivated biologists for some time is that these guys have this really, really simple nervous system. And the neat thing about their nervous system is they've got a brain. I have messed up how I'm presenting to you. So usually I do this from the iPad, and for some reason my iPad didn't connect. So if you give me just a second, I will have a laser pointer, which is going to be important. There we go. Haha, beautiful. So here's the brain. And if you look in here, you can see all these little dots. Each of those little dots is a neuron. Okay, if you were to count them, there are about 2,000. So it's not that many neurons. And then over here, this is a ganglia. The leech has 21 of these ganglia, one for every one of its 21 segments. Those ganglia have about 400 cells each. And the great thing about these cells, and I'm going to outline one here. You can see how big that is. Even the little ones, here's a little one. The little guys are maybe, 50 to 75 microns across. The big ones are 150 microns across. So people always ask me, well, why would you study leeches? It's because our electrodes that we put into those cells to record from are about 4 to 5 microns across. So getting that into a 5 micron diameter mammalian cell is really hard, but getting it into a leech cell that's between 50 and 150 microns, it's like parking a Fiat in a space for a semi. It's not that hard. So we like to use this system because it's very easy to record from these neurons. All right. So if you did the math, and I'd be surprised if you guys didn't, kind of knowing your work, right? We've got a brain with 2,000 neurons. We've got these ganglia with about 400 each. And that's about 10,000 neurons in the entire animal. When you think about that in terms of animals, that's actually incredibly simple, right? If you think of a cockroach, they have about 1 million neurons. You have as many neurons as there are stars in the night sky. So you kind of think about what can you do with 10,000? That's not that many. But even though they only have 10,000 neurons, these animals are capable of finding food. They're capable of finding mates, escaping, and moving, like locomoting around their environment. So what has kind of driven my whole career is looking at animals with these just teeny little brains where they're not working with much and saying, okay, how can you do something complicated with that little neural real estate? So I'm going to focus on some work that I did more years ago than I like to admit right now, which is about how leeches find prey. And then I'll have a part 2 for you that's the more recent stuff about movement. So when we started thinking about how leeches find prey, we started thinking about what stimuli are they paying attention to. They live in the water the majority of their life, or at least this species does. And in living in water, it's going to be able to sense water movement, but it's going to have to separate sort of like useful water movement from water movement that isn't interesting. So basically normal, natural stimuli that are not tasty from something that it wants to eat. How does it do that? Well, we started thinking about using frequencies of waves. And what we found is that some frequencies are easier for them to localize than others. So literally, I just had this bobber bobbing up and down in the water, and I looked at whether or not they were able to find where that originated. And you'll see right here at about 8 hertz. That's where they do their best localizing. Either side of that, they don't find the stimulus, they're probably not interested in it. But then I found this really cool paper that kind of made me question my existence for a minute, as really cool papers tend to do. And I found that these guys switch prey as they get bigger. So the little ones, because they have little jaws, they can't bite through thick skin. So they tend to feed on frogs. And then as they get bigger, they start feeding on fish. They get a little bit bigger. They start feeding on birds. And then they get bigger than that and they start feeding on mammals. And they're actually not reproductively viable unless they're feeding on mammals. So if my theory holds true that they're separating out the signal from the noise using frequency, then as they change the prey that they're looking at, they should change frequency as well. And that's exactly what we found. So this is looking at a juvenile leech, so like in the frog range here. And this guy is paying attention to a 2 hertz stimulus. So that's kind of fun, right? There's this shift. And then we started to ask all these questions about where that shift came from, right? And that led us to think about water. And what are the relevant parts of water movement? Well, part of it, of course, is the mechanosensory. It's that the waves are moving something. But there's also another component. If on a sunny day you throw a rock into a pond, you're going to see bands of light like this. And that's because that water is creating lenses on the surface, or the waves are creating lenses on the surface of the water, and they're focusing that light from above down to the bottom. And we thought, what if leeches are paying attention to the visual elements of this? So we said, do they have eyes? Right? They do. They have 10 eyes on their head. So here are these little black spots. Those have between 50 and 10 photoreceptors, depending where they are on the head. And then we have these multimodal cincilla. These are wrapped around each of the 21 segments, sort of like a hula hoop. And they have 14 per segment. And those have mechanosensory hairs on them. This is under a TEM microscope. And if you were to section below that, you'd find a couple photoreceptors. So I always tell people they have 294 of these mechanoreceptors and 294 simple eyes and 10 complex eyes. In other words, they're going to start something. So in untangling this, we had to say, well, how do we make a solely visual stimulus? And we came up with this idea of abstracting out what we saw, quite literally putting a piece of paper under their arena and making waves. And we turned it into a digital video that we could project with like the same kind of projector you have in a classroom. So this is our video, and there's an origin of the stimulus, and then the waves would be moving this way. And what we found is that when they're only looking at visual stimuli, they are orienting towards that 2 hertz. Okay. So it doesn't matter if you're adult or a juvenile, everybody likes 2 hertz visually. When you go to mechanosensory, now we decided, all right, what we're going to do is we're going to turn out all the lights. We're going to put the critters under infrared. They can't see infrared, and we're going to see what happens. So that is what we did. And we have this thing making waves in the water. And what we found is that whether you're juvenile or whether you're an adult, you're looking at about 8 to 12 hertz. So lower frequency if it's visual, higher frequency if it's mechanosensory. And that got kind of interesting because this curve also looked like an adult. So we went a step further. And this is like, we called this the stimulus battle royale. So we said, all right, down here, we're going to have our bobber that's bobbing up and down, making the mechanosensory stimulus. And then we're going to project that visual stimulus so that it thinks it's over here. And we're going to see how the animals locate it. What do they locate? So basically, if they have two stimuli going on and they're conflicting, which one do they go to? So your choices here, and I'm kind of walking you through it because this figure has a lot of stuff on it, is there's no stimulus. There is a visual stimulus on its own, that'll be in red. A mechanosensory stimulus, that'll be in blue. And a multimodal, that means it's mechanosensory and visual in the same location. In different locations going on at the same time. What we found is that for both of them, they could localize the visual stimulus. They could localize, so down here we've got finding the mechanical stimulus. They're more likely to find that if that's the one that's working. And then this is the neat part. When you run the visual stimulus and the mechanosensory stimulus, the babies go to either. It's like a 50-50 shot which one they're going to go to. The adults go to the mechanical stimulus. They very rarely find the visual. Okay, so that tells us that the adults are mechanosensory animals and the babies are a little more, they kind of take in everything. How does this happen? And it turns out that there had been a study done before I ever did this research that found that over the course of a leech's life, it develops more mechanosensory hairs on those cincilla, so those little receptors all over its body, and it develops more neurons going out to those cincilla. So basically the system is getting more sensitive and it's getting more sensors, okay? And in doing that, that biases it. If you think about it, it makes that mechanosensory signal louder to it. So it washes away the visual. I'm doing some work with a group at UNC now, and we're working on some computer models of this, which is really exciting. All right, so that's the first part. Basically, we figured out sort of how the animals are receiving information from their environment just by looking at their behavior. This is part 2, which is looking at much more, how do they get to these stimuli? All right, so we're focusing on locomotion. And one of the cool things about leeches is they've got 2 modes of locomotion. And these are easily separable with no training, all right? This is swimming. It's this beautiful sinusoid. In fact, Leonardo da Vinci wrote about it and was very captivated by it. And the other mode of locomotion is crawling. Crawling is, uh-oh. Come on, Cindy. Hold on, I did something. There we go. I got too excited and I like touched the video. Oh, go back. Oh, geez. All right, there we go. Let's see if I can get this to go now. It might not work. It's okay. All right, it's not working for me. That's fine. Crawling is an anterior to posterior elongation followed by an anterior to posterior contraction. So basically they stretch out and get really skinny, and then they put down their front sucker and they start to contract. And that's kind of how they do it. And people found this interesting. And remember that in this system, we have these massive, massive, massive neurons, okay? So now you've got this system that has these 2 kind of cool behaviors. They've got a bunch of neurons, and both of these behaviors take very different means of control. That, to a neuroscientist, like, that's what we live for, okay? Add into that, that when you look at these ganglia, it turns out that the cells are in very similar places, ganglion to ganglion. So basically the cells in this ganglion will be very similar to the ones in that ganglion. And from animal to animal. So we have a map. And to give you an idea of this map, like all these cells marked P here, these are pressure sensitive neurons. I know if I record from that neuron and I touch the animal's skin, I'm going to get action potentials. So it's very easy to find them. The most you're off by is a single cell. It's pretty cool. Okay. Now, what have I done? I've done something rather horrible to my display here. Oh well, we'll just keep going. All right, so it gets even cooler than that. Now we've got voltage sensitive dyes. There's Kevin Brigman and Bill Christon get together. They dump voltage sensitive dyes on these critters, and they dump them on one of the ganglion while the rest of the body is still attached. It's very science fiction. And they look at this and they find when the cells are active based on what those dyes are doing, because those dyes glow when the cells are active. And what I want you guys to notice is that different cells are active at different times, and that you've got some cells like this one here that are active in both behaviors. So they can even sort of reconfigure the circuits that are controlling these behaviors. Pretty cool stuff. But there's one big question that no one asked. And that big question is why? Why do you have two modes of locomotion, right? I wanted to know that. Why is it important to have two? Why not just like do one really well? Now I'm starting to sound like my mother. All right. So when you go back to the orientation videos, I'm going to show you two separate orientation behaviors. And I want you to pay attention to what the animals are doing when they're orienting. All right. So in this one, I've got it orienting to a shadow. Leeches love to be under stuff. So the first thing we actually looked at them doing was finding shadows. And they get lost. They've only got 10,000 neurons. You got to give them a little break. There it goes. And I should say, so that I don't give you guys nightmares, these videos are sped up a lot because they're really slow. We'd be here all day if we just watched them moving normal speed. Okay, so my other video, this guy is orienting to what we call our prey stimulus. So this is the thing poking the water. Little atmosphere added too, you know. Gets me every time. So I want you to think about what they didn't do. I said they can swim and they can crawl. That was crawling. Both videos. Can they swim in this arena? Sure. Here's the video of them swimming. So they can do it. They're just not doing it. And that's interesting, right? Why do you pick one behavior over another? So we started looking at why are they crawling so much? And if you look at their behaviors under normal conditions, what you're going to see is that leeches spend, this is normal conditions, no stimulus. They spend a certain amount of time crawling, a lot less time swimming, and their favorite thing to do is nothing. And they do that about 70% of the time. When you add in a stimulus, then what happens, and we're adding in our prey stimulus, and these are hungry leeches, so they're looking for it. Then what you see is that they crawl significantly more than they did before, and they do nothing significantly less. They're not swimming more. So we said, well, what's so important about crawling that they're doing it so much? And we got this idea of providing what we called conditional stimuli. And a conditional stimulus was we gave them a stimulus only when they crawled, only when they swam, or only when they did nothing, and looked at how good they were at localizing it. And we found that they were really good at finding the stimulus if it was given while they were crawling. The second best is when they're doing nothing, and they're really bad when they're swimming. Basically, it turns out they like turn off their sensory receptors because when they're swimming, it's overload. So that kind of interested us because we're like, well, now you've got to decide to crawl, and you've got to have this sensory information come in and like flip a switch in your brain that says crawl more. So that got me interested in the brain. And it turns out that Karen Mesce, who was my advisor for my second postdoc, had found this really cool neuron. And the neuron's R3B1, so located right around in here. And that R3B1 is a neuron that decides whether or not the animal swims or crawls. In fact, what Karen found is she could change the behavior by changing how she was stimulating the neuron. So if you look here, you'll see the motor pattern. It's a little slower. I always think of these making sounds. So I would go shh, 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 and that's opposed to shh, which is crawling. And she found if she gave it one stimulus frequency, she'd get that shh, shh, and the other stimulus frequency, she'd get crawling. So this neuron somehow biases the animal one way or another. It's like a little toggle. So, okay, these animals can not locomote, they can swim, they can crawl. In normal conditions, remember we found that they non-locomote the most, and then crawling is number 2. What happens if we get rid of the brain? So we said, what if we get rid of that R3B1, that cell that tells them to swim or crawl? And so we kind of just went in here and we gave them the politician treatment and took their brain out. All right. We found this. Now, all of a sudden, there's no crawling. It's gone. And swimming has increased by a lot. It used to be down here. Now it's way up here. That's interesting. Some of these animals swam for an entire trial. So the whole time we're looking at them, they're swimming. It's amazing. And we said, okay, what happened to crawling? And around this time, you know, you go away, you go on break, whatever, you come back, you kind of get out your animal that didn't crawl, and you're all excited to show your friends. And I'm going to show you normal crawling. This is normal. I've colorized it so that purple blue is where the animal's getting thinner, that yellow white is where it's thicker. So you can kind of see it's getting skinny front to back and then thick front to back. 46 days later, you look at your non-crawling animal, and your non-crawling animal looks like this. Just perfectly normal. And you're like, okay, it's an invertebrate. Invertebrates are kind of weird. Maybe there was like some regeneration stuff. No, there was not. So there's still a big old gap between the brain and that first ganglion, just where I cut it, 96 days before this picture was taken. Things didn't regrow. So now what? How is the nervous system reconfiguring to suddenly crawl again? And the first thing we had to do is figure out the timeline. All right. We figured out the probability of crawling on any given day. The first day that they have any likelihood of crawling is day 4. Okay. And then by about day 10, almost everybody's crawling. But if you look at the volume of crawls, even at day 10, it's still really low. It's not until about a month and a half after their surgery that they're back to what we would call normal amounts of movement. And then when you look at the crawls themselves, they look like this. It's like, they look like gum that you poured on something. They're not these beautiful elongating leeches anymore. That was 6 days after the lesion. So now let's go 10 days out. And 10 days out, still, and this is actually a pretty good one. right? That's not good. That's limping at best. So we started to think about how do we get to the really good crawls faster? And the idea was if we can speed up that recovery, we can figure out how it's working. And this kind of corresponded with a debate that I was having with my advisor at the time of, I'm from a sensory background. I've always studied sensory systems. I said, it's sensory. It's got to be a sensory issue. And she said, no, it's motor. She came from a motor background. And so I said, okay, well, we'll do PT and we'll figure it out. And that was how the story began. So we came up with these 3 goals. The first one is decrease the number of days until they start crawling. Number 2, get them to crawl as much as they did before. And number 3, get them to crawl properly with really good, beautiful coordination. So the first problem is, remember, I told you they love doing nothing. That's like their favorite thing to do. And that's okay, I identify with that. So we had to figure out how to get them moving. And we were able to use some work by a colleague of mine who figured out they really don't like UV light. And I'm gonna kind of skip ahead because I'm being long-winded today. But basically we figured out if we used one of those big UV lamps that you use over a fish tank, we could get them to crawl many, many, many times more than if we had no light at all. So if they could crawl, we would see it. And that's always the thing you worry about, right? You worry, maybe they can do it and maybe I can't see it because I'm not looking at them at that time. So then we thought we need to create physical therapy, and we kind of started looking at the worst animals in terms of figuring out how to make them better. So when you look at this video, I'm going to try to point out where this guy is contracting. So there's a contraction here. And this one's really bad. There's like something going on there. Something there, a little more up here. Now we're back here again. So you're kind of getting this idea that rather than having that anterior to posterior wave of contraction and anterior to posterior, or wave of elongation, anterior to posterior contraction, it's all just happening all over the map. All right, I'm gonna let this one finish because I do feel like once I get him started, I gotta let him go. There he goes. And he finally gets it. And we said, okay, so in an abstract sense, what we have is this, where each segment is contracting on its own and it's not talking to the ones in front of or behind it. So what we need to do when we're making physical therapy is we need to make a way to activate those from front to back to train the proper order. So this. How do we do that? So we came up with the idea of squeezing them into the shape that they're at. Remember, when they elongate, they get skinny. So we're like, if we force them to be skinny, like put a corset on them, then they are going to basically elongate. So we made these little silicone molds. This is 3D printed. I've got a 3D printed leech mold that I pour silicone over, and that creates this device. And then what we did is we created a leech standard curve. We figured out how thick leeches of different weights were so that I know this is a 3-gram leech. If it's a 3-gram leech, I'm gonna put a 7mm squeezer on it, right? It just tells me like its size. And then we put that on a treadmill because we wanted the device to just feed this critter through that squeezer so that its segments would work in order. Okay. And I've got a little video of it working here. And eventually you'll see his little head come out. There it goes. And as this guy's going through here, there's a skinny part. Whoops. It didn't stay on the video, but it's getting skinnier. So we got really excited about this. And one thing, I always take this part out and I put it in because I knew you guys would laugh, and I don't know, we need that nowadays. Oh, we do this, I put the 10 times in, we do this 10 times. They have to crawl 10 steps. So we had to make this thing move at injured leech speed. And I put this little chicken here because he stays on there better than my leeches. And I just wanted to show you how horribly slow that is.
Is that like the sound for Top Gun?
It is Top Gun. I needed something appropriate. They're wickedly slow. But that's kind of the fun of making the treadmill, was that we had to make it just the slowest thing ever. All right. So I can move on from there. So now I know what you want to know. You want to know, did it work, right? That's what we wanted to know. And so we're like, okay, is the first day of crawling earlier? No, it is not. Air goes out of the room, right? Like, oh, no. Is there more crawling? No, there is not. There might be a little bit more on day 6, but it's not really anything to write home about, right? Okay, maybe the crawling's better. You know, at this point, we were really upset. So we said, how do you assess crawl quality? We realized that one of the differences between the normal guys and the recovering guys is this guy really elongates, and when this recovering guy is elongating, it doesn't get nearly as long. So we said, okay, we're gonna look at how stretched out they get and basically see how big the steps they're taking are. And we said, okay, they're better. There's gotta be something good about this treadmill. I mean, after all, it's the most technologically advanced leech physical therapy device in existence. And this is looking at that length ratio before surgery. They go to about 2.25 times their lengths. With no therapy on day 7, they're at about 1.5 times resting length. And after the treadmill, they're at about 1.5 times resting length. It didn't do it. So around now, we are upset. We're dejected. We're thinking of giving up research, right? That's how it goes. And then you think about, why didn't it work? And it's because this is not hiking. In this case, much like our treadmill, there is a machine doing everything for this woman. She's not getting any physiological benefit out of riding a Segway through the woods. So we said, okay, what if we make them crawl their own butts through this thing? And so now, instead of mounting the squeezer to a treadmill, now we're just holding it down on top of them. And they still have to do 10 steps through the squeezer, and then we test them after that 10 steps. And we wanted to see what that does. I had to put the times 10 in there. I always forget to tell people it's 10 steps. So when we do that squeezer, you look for the first day of crawling, and okay, maybe it's like a little earlier, you know. We kept saying we need more data, we need more data, we need more data, and this number has just held. When you look at the volume of crawling, how much crawling they're doing, it's not significantly different. So about now, we're getting really worried. We're like, oh my God, we failed again. And then we looked at the quality. And when they're doing that static therapy, they're way, way, way better than if they get no therapy. And these guys were all tested on, this is all day 7. So they're all tested on that same point in recovery, which is a pretty early point. And these guys are actually no longer significantly different from before. That's pretty cool. So now we're getting quality, but not quantity. All right. So they said, okay, we want quantity. We want to have them crawl a lot. We want to have them resume their normal crawl cycle numbers earlier. And around then I had a student that said, well, they can't crawl and swim at the same time. So what if the swimming is disruptive because they're swimming a lot after this injury? I said, okay, let's figure it out. So we take these guys, and instead of putting them in a little container that allows enough water for them to swim, we put them in water so shallow, all it does is keep them moist enough to keep them alive. Okay. So what happens in that case is that the first day of crawling decreases significantly. So now, instead of crawling for the first time at like day 5, these guys are crawling for the first time at day 3. Yay, right? We're getting somewhere. This is exciting. A lot of them actually crawl on day 1, which is really cool. So then we look at the volume of crawling, and you can see, I always joke about the bloody obvious test with data. This is, to me, pretty bloody obvious, right? They're crawling more. They're getting back to their pre-test values by 10 days following transection. Remember, it used to take them 48. So I'm doing good. All right. Now, one of the things I want to stress to you is that if you put them in shallow water, this doesn't on its own cause more crawling. So if we house them in shallow water and they were never transected, they don't show an increase. You only see the increase in the transected individuals. So it's something to do with what the nervous system is doing while it's being housed in that shallow water. All right. Is more better, right? Did we get the trifecta? Did we get good coordination? No, we did not. And some of them are definitely crawling better. I think that's because they are crawling. But when you look at the mean down here, our mean is no better than if they had no therapy at all. So basically they're crawling a lot. They're just not doing very well. So now we've got the opposite problem from before. We have quantity, but no quality. Okay, and this is what one of these looks like. And what I want you to see is that this guy's gonna push himself forward with his butt rather than elongating sort of front to back and contracting front to back. He's gonna push himself forward with his rear end. You can kind of see that where it's like, it's not good. And now he's gonna start from part way up his body. So there isn't that complete coordination on these. Okay, so quick summary, and then I'm gonna tell you how it works. So we decrease the number of days until crawling onset by putting the leeches in shallow water and just housing them in that during the recovery process. We resumed the normal number of crawl cycles by putting them in shallow water. And we resumed normal coordination by using that squeezer and making them crawl themselves through it. So can we get the best of both worlds? And what we've been doing, and we were kind of cut off of this by COVID, which is frustrating, was making them walk through the squeezer 10 times and then housing them in shallow water. And what we found was that they did show an increase in crawl quality, and they did have more crawl quantity than the guys that just did the squeezer, but they're not quite as good as one of those on their own. But here's one crawling, so I've always got to show them off. They're pretty cool. And this one doesn't latch with his front, so he slides backwards, but you can see that he's got that really nice coordination, anterior, posterior, anterior, posterior. Okay, so your big picture, let's zoom out, mile high view. Recovery takes motivation and ability, and these could have different mechanisms, okay? What I'm gonna call motivation is the number of, like the amount of crawling you do, how early you do it, how much you do it. And we know in leeches that decision to crawl can be biased by dopamine. If you put dopamine on a nervous system, it'll crawl. If you put serotonin on the nervous system, it'll swim. So what we think is going on sort of under the hood is we think that they're putting on more receptors or secreting more dopamine, and that's sort of biasing their system. and that's what's happening by keeping them in shallow water. Now for ability, what we think we're doing is we think that this sort of squeezer thing is entraining their system. But how would it be entraining their system? It turns out that there are stretch receptors that are located in each ganglion, and those stretch receptors feed into the motor circuit. Okay, and there were hypothetical sort of connections between these stretch receptors. So he said, well, maybe what we're doing in squeezing them is we're like poking at the stretch receptors. We're poking at the sensory part of their skin that's saying, yeah, you're squeezed. This is what a normal stretch receptor looks like. So this is a neuron that's out in the skin, and it's sending its projections into that ganglion, okay? And you can see it's only on one side of the ganglion. Now, this is a stretch receptor in a recovered animal. And what I want you to see is it's going to the ganglion above it and the one behind it. So now what we think is going on is we think the sensory system is filling in that gap that's left by the brain. Brain's gone, so now sensory is going to cover it and sort of give the information that's needed to develop that coordination. And we are facilitating that by making those neurons active and making them active in the right order. All right. So your really big picture from these sort of two stories is that the brain is using sensory information to direct the decision to crawl. And that's actually really important because it makes that animal better at locating things. And then if we get rid of the brain, we can recover that ability to crawl, but that's going to take motivation coming from dopamine, we think. And it's going to take ability, which we think is coming from the stretch receptors growing in and coordinating the adjacent ganglia. And so this is like my army of undergrads. I'm in an undergrad institution and we've been doing this for a while. So I just have to give them some props because they are amazing students and they put up with my crap. All right, with that, any questions?
I have a lot, but yeah, I do. Look, it's really fascinating that you can compute that much with 10,000 neurons.
Right?
Right? So wait, let's first, about the students though. So do any of you have a question for Cindy?
I'm trying to get used to
I have a
question who's
question
just
from the beginning.
had it an animal and trying to recover it. Go, Vitor.
Faça a pergunta, Vitor.
Video cuts a little bit. I don't know what you were saying.
What?
Hello?
Oh, hi, I can hear you.
Okay. So it's from the beginning when oh, it's my connection. It's unstable. So let me change here. So Fernanda says something. I will change my internet.
I really enjoyed it. I think it's very cool that you are so excited about it. It's just crazy.
It's been a lot of fun, and I will tell you, it has not gone once how we thought it would go. And we literally, when that treadmill didn't work, because I think that was 2 years getting the treadmill, and like in the thought process, we were working on other things in the lab at the time. And when it didn't work, we were heartbroken. And then once we figured out that they needed that push, they needed to be doing it themselves, I was like, oh, so if you're not involved, you're not going to learn anything. And then, you know, I spend most of my day teaching, so I was like, duh. And so I think it's a very satisfying, very satisfying piece.
Thank you very much. It was awesome.
You're welcome.
Okay, so vai, Vitor, faça a pergunta.
About the beginning of presentation, when you put the first graphs about the frequency of the stimulus, the light stimulus, I didn't understand if you were tapping the water and lighting. I didn't understand if it was something simultaneous.
Yeah, so with with the mechanosensory one is just this thing that's pushing the water. And then when we wanted to do visual, the first visual stimulus we did was I took one of our arenas, which if you've been in a snowy area, it's a saucer sled. It's something we slide down the hills on. I cut the bottom out of it and put plexiglass on the bottom. And we put that above the one that the leech was in. And we tapped the water in the one up top.
Ah, okay.
And it turned out the lensing was good enough. And then over time, we developed the digital, sort of the abstract stimulus where it's just bars going through.
Okay, thank you.
No problem.
Because I was a little bit, I was thinking about the frequency itself. Because the frequency, it's a low frequency, 2 hertz is something really low. It has to do with the nervous system that I don't want a lot of stimulus, but I have to a little bit. I don't know if something like this.
Yeah, I've been trying to figure that out. And, you know, that's been one of our questions from the beginning is what are these frequencies? But I think about, you know, the frequency of a frog swimming, and they kind of stroke and glide and stroke and glide. And my hope is that that is that low. The truth is I don't know what these are related to in the real world. And one of my random things I've wanted to do is just get a bunch of scuba diving equipment and just kind of sit at the bottom and like take videos of some different creatures and just figure it out, because it's very curious. And one of the things that's always been really curious to me, and this is why we're simulating it now, is I can't wrap my head around the fact that these animals grow from the juveniles we used were 0.1 grams and the adults were 3 grams. So like hundreds of times their body weight and length. And they're still able to deal with the fact that their sensors, the sensors have gotten further apart. So how do you still see the same stimulus? And I've never been able to wrap my head around that.
I have a couple questions about the frequency part. So you said that as it becomes bigger, it starts to prey on larger and larger animals, right? But
no mammal, we don't know larger.
Okay, but I mean, you said like frog and fish, then duck, then cow,
I always
right?
like to say cow, but I think really it's probably muskrat.
Yeah, well,
Yeah.
because I'm thinking that the bigger the animal, the lower its natural frequencies should be. But you're seeing the opposite effect. So the larger the leech, the higher the frequency it's most predisposed to detect.
Well,
right?
so one thing I was thinking, and like with fish, when they're just hanging out, because what the leech really wants is it wants an animal that's still.
Okay.
A fish that's just hanging out isn't really moving much, right? They just kind of. But maybe like our original thought was hoof stock cows and deer, and they're going to be licking the water.
All right.
Right. So that's actually a little faster, even though they're still. But again, I mean, this is like where I just need to learn how to scuba dive and then get some underwater camera equipment and just get a GoPro and boat for a while.
Because I mean, I had a bunch of thoughts about what these frequencies could be.
Really?
Yes. So for instance, they could not be like the active, the animal being active, but rather you have like these environmental fluctuations in the water and they refract on the animals. Like a point source refracting light and different lengths, different sizes are going to reflect light at different intensities for different frequencies. So if you have like just a random noisy environment and you have like a small ball, a big ball, a bigger ball, it's going to scatter these random fluctuations with different intensities, with different frequencies, just like, I don't know, particulates in the air scatter sunlight at different rates so that you can have a blue sky and a red sunset.
Got it.
So, And so I just thought it might have something to do with it, like the characteristic frequencies of different sized perturbations in the water. Because I was really thinking that the animal is like, it's still, I mean, the leech is not chasing after the animal. So if it's moving, it's not really likely to get bitten. So it must be lying around somehow. Does that make sense to you?
A bit. It's an interesting, it's like one of those things that I don't think we've ever considered. I did think about stimulus directionality, that if it's raining, that's not going to have a direction, right?
Yeah.
But we're trying to give something directional. And what I didn't talk about is the really high frequencies they don't like and they go away from, which was
which might be something chasing after them or something active, right? I mean, they want things that are still and quiet and non-threatening. I mean, I'm completely speculating here, but I think it's really exciting because since it is a really simple nervous system, it cannot be taken too much of a sophisticated input. So it must be something simple it's detecting and running away from. Which leads to my second question, which is, so if you have something in the water, you're not going to have like one frequency, you're going to have a spectrum. So do you have any results on its response to multi-frequency stimuli? So you have like two frequencies at the same point, but with different frequencies, just like you have a regular spectrum of anything. And perhaps it's like submarines detecting one another. You have like this spectrum characteristic, one submarine, oh, that's a frog, that's a fish, that's a cow or something.
Yeah, we don't. I mean, we got some data early on that made it look like the harmonics was interesting. But we didn't do a lot with that. And one of the things I want to do, and again, this is kind of where the simulation comes in, is what we really want to do is figure out how much the simulation so far has shown that the processing is, as best we can tell, the simplest thing we could come up with. And one of the questions is how much noise can it handle? And so what I've got the student working on that work doing is trying to figure out what trips up his stimulus. And then if we give that to the animal, are we going to have animals that can't localize something? So, because I've always had this question of how much noise can you deal with?
Right.
And
well, that's one of the things that being able to detect multiple frequencies might help, because the white noise, if you just look at one frequency, white noise and like coherent noise are indistinguishable. You just have this amplitude. But you have like multiple frequencies and you see a spectrum cell, that's flat one's white noise, but this peak one is something else. So you can actually pinpoint that other thing.
One thing we think they were doing, and this came out of the simulation data, he would come up with this, they had this really, because he simulated the adults, but they had this really weird low peak. And we said, well, wait, what if you invert that low peak? What if they're sensing it, but they're like, nope, I'm not going to go towards it. And he inverted it, and all of a sudden it's like, it was my data. Like you can put them on top of each other. It's really neat. And so we're kind of just, I think that that's part of it, is that there are certain frequencies where they're like, no, like this chunk of it is not good. This chunk is good. This might be interesting. And I think they're kind of going between that. What I want to do eventually is record from the sensor and figure out what the sensor can do. I mean, that's really what you need to do. Right? At some point, is just like get in there.
Yeah, okay, I'll move on from the frequency part because I can talk all afternoon. So 2 questions. Why are the neurons so big? I mean, in mammals, typically bigger neurons means they have more connections. But I'm sure that's not the case here. I mean, you have fewer
myelin.
Yeah, you don't have myelin, but I mean, you have fewer neurons in that animal than the average number of connections of pyramidal neuron in a human cortex. So they're not having many connections. So why are they that big? Is there no myelin thing?
It's a lot the myelin thing.
Right.
Since you don't have myelin, the way to make your cells function in a specific speed is to have them be bigger.
But so just mean bigger axons or bigger soma as well?
So they have bigger somas. Like everything's bigger. So it's just this sort of, it's the solution to the same problem, right, of speed. It's just a different, the different way of going about it. So yeah, they have those big, they're so big.
Yeah, I'm looking at them and saying, wow, that's...
The cells are pretty well connected too. They have some pretty complex connection patterns. And one of the studies I was doing was looking at, like, we thought the gap junctions were being adjusted. So these like electrical connections between cells, we thought they were like closing, opening, depending on what hormones were present. And we started kind of playing with that idea. And then abandoned it because other stuff got more interesting. But the cells have these really complex relationships between each other, despite not having that many. At the same time, it still makes them a great animal. And when I saw big cells and 21 chances in the same animal to hit the cell I wanted, I was like, yes, I can do that.
Right. Well, I think even I could do that because, yeah, I mean, even me with my poor experimental skills.
I sometimes don't look through the microscope and you just do it. You like, I've got a
eyeballing it
that makes a sound when I get in a cell. It goes,
wow.
And so you just kind of like pull out and you're like, oh, I'm not in the right cell, move over. Okay, I'm in a cell. It's fun. Okay. You can literally do it by ear.
Well, that's impressive. Okay, one more question. After net start them, apparently after some time they actually learn to crawl again and be their normal self. So why do they need their brains? You know that story about that corals, when they are young, they have brains, and then they settle down, they eat their brains, and like one of the 2 species that eat their own brains.
I didn't know that.
Yes, and you know what the other one is? Tenure professors. And they separate their brains.
I thought you were going to say Donald Trump.
I was about to start with, come on. Yeah, I mean, it's something to eat to begin with. But my point is, what do they need the brains? I mean, apparently they can live after some training, especially with some physiotherapy. After a while, they can live without it.
Well, I think, you know, you can reestablish a connection, but one of the things we're interested in is we don't think that the reestablished connection, like, they can crawl again, but what can they do in terms of complex behavior? Can they like crawl up walls? Can they hunt? Can they search?
So it's less adaptive.
I think it's like the higher order stuff, and I mean, we do this too, right? The higher order stuff is up in the brain, in certain regions of the brain, and anything that we can like throw into the spinal cord or the brainstem and just have it like go on automatic, you know, that's what we do so that the rest of our brain can think big, beautiful thoughts. And I think that that's kind of what their brain does. But their nervous system is interesting in that if you look closely at the brain is evolutionarily 3 ganglia that just squish together. So it has its roots in that. And I think that's why they're able to kind of reconfigure ganglia, is that there's not so much difference between the ganglion and brain.
And how much do they learn? I mean, you talked about dopamine, and that's related to the one movement that they do when they're actually trying to adapt to some outside stimuli. So to that extent, can they learn some behavior and become more adaptive?
There have been some people that have looked at like classical conditioning and that kind of stuff to see if they can learn. I've heard there's been evidence of it, but I imagine it's pretty slow. I do think that what we notice when we're training them to crawl is that after an exposure, they kind of get it. So I think that there is some learning. How they do it, we don't know. And that's the neat thing here too, is it's possible just by using that squeezer that we're causing them to secrete dopamine. It's possible that we're tapping into something intrinsic to them. We don't know. So one of the plans is to like draw out some of their blood and test it and just see what the blood tells us, or did they have more dopamine or no? And it's just hard to get their blood because they have an open circulatory system. So it's kind of a pool of everything in there.
Oh, wow. Oh, that's so cool.
Yeah, and one thing we want to do, you'd love this. So with dopamine, right, you can find a lot of drugs. There are a lot of drugs on the market right now that modify dopamine and serotonin levels, and leeches have no blood-brain barrier. So we can grind up the drug, put it in their water, and have them swim around in it. And then just see what happens.
I'd love to see you try to explain that to the police, you know?
Right? I needed the drugs for my leeches.
Yeah, it's for my leeches, not for me.
Right.
Oh, that sounds cool.
But then, and we've always wondered, like, what happens if we just drop a couple antidepressants in the water? You know? Maybe they start dancing. We don't know.
Well, that might be the like evolutionary origin of dancing. You know, perhaps it's more basal than we thought. That's cool.
But then, which one's the dance? Is it the crawl or is it the swim?
No, that's a combination of both. I mean, you're gonna have like a slug rave or something.
Well, we do always have music blaring in the lab. So.
Oh, by the way, a little bit of trivia about Top Gun. You know that there's an actual Top Gun school where they teach fighter combat tactics, right, for the U.S. Navy.
Yeah.
And apparently, if you in that school you're a student and you actually quote from the movie, you have to put $5 in a jar. It's like a swear jar, but for quotes from Top Gun.
I love it. I love it. Yeah. Well, when we got that treadmill all set up, and we figured out how slow a lesioned leech moves, and kind of went through all this stuff. When we finally turned it on and got it moving, because originally it moved pretty fast, we're like watching it and just, you can't help but laugh because it's just so painfully slow. And have talked about even making that more fancy by putting in like a brake beam so it adjusts to the leech's speed. Because it's all Arduino.
Right.
It's pretty groovy. So we can expand it if it worked, you know.
Well, maybe we can say a leech physiotherapist. That's really a nice thing to put in your CV.
I think that's my Twitter handle, like leech physiotherapist. Yeah.
Oh.
I think Twitter, LinkedIn, one of those like social networking sites. I was like, I'm just going to put it on there, like leech physical therapist, and people can think what they want.
They have the leech, they can bring it to you and see if you have like an inspirational movie about
right. I mean, you're like a professional zombie, so.
Yeah. Have you ever seen my zombie presentation? Maybe?
You showed it to me. I think you had a poster.
Yeah, the American Physics Association meeting in Denver.
Yes.
Yeah.
You sent me one, but I feel like you snuck it into a conference.
No, I did. I mean, it wasn't a program. I did not like smuggle it in.
I thought that was the earlier one where you like smuggled it in and like.
Did I do that? I mean, I can be subversive sometimes, but usually I'm.
No, it's like neurosciences, but it would have been neurosciences like.
Look, I did give a talk about zombies last year in Newcastle, and it's cool because I mean they invited me to give not that talk. They invited me to give another talk, and I gave this other talk, and there were like a reasonable number of people in the room, and it was fun and all. And then I thought, okay, I want to give another talk about zombies, and they advertised that, and there's no chairs left. The people like. Sprouting out of the room from every floor in the building. Want to know what this zombie population dynamics was about? So that was cool.
Well, you know, you've got to do that. If you're not having fun and you're not like thinking about some of this crazy stuff, then what are you doing, right?
And the math is real. I mean, it's real proper math. Some of it may even be useful at some point. So
yeah, well, it might be useful with the current like.
Well, yeah, but look, it's like this. You have models like this for prey and predator, or prey and I don't know the name for that is prey, but like parasites and prey, whatever. So zombieism is just one different type of relationship where you have an encounter and either between two populations, and either a member of a population is destroyed or a member of the other population becomes part of the initial population. So it's just like mathematically, it's just terms and equations. COVID models are simple like that. I mean, if you're infected, you can contract COVID. If you come close to someone infected, you can contract COVID. What happens is that you not kill the COVID victim if you're not. That's the main difference.
Yeah.
But mathematically, it's the same
You're not
thing.
infected and actively seeking people out and like.
Yeah, do you have COVID and start blasting them to kingdom?
I don't know. They might be. I mean.
Well, that might be even a prophylactic. I'm not sure. I mean, I wouldn't recommend it. But yeah, so it's fun. But anyway. So I think, I mean, we took enough of your time. It's like 1.5 hours, but thank you so very much. I think everyone here really loved your presentation. It was an awesome presentation. The subject's really fast.
Hey,
So,
thank you. It was fun to talk to you guys.
So one thing I'm going to ask you, if you have like the papers that, your papers that you wrote about these subjects, can you just send me a link and I'm going to post it to them so if they want to follow up?
Absolutely. I don't have the physical therapy stuff is one of my jobs was supposed to be to write it up. And right now I think we are just short on the data we needed because of this stupid pandemic. But yeah, I'll send you, I think it's all on my website. So I'll send it to you.
Good. Awesome. Thank you so much, Cindy. So do anyone else have any questions for Cindy? Okay, that's plenty myself. Anyway, so thank you again, Cindy. And I really hope we see you again.
Yeah,
Hopefully.
I would love to. Eventually, right? Like when the world stops ending.
Yeah. We're gonna talk again. We're gonna go to Chinatown again. Remember last time we were in DC.
Oh, that was so.
Yeah, that Dutch place.
I just keep going back to New Orleans though. That was the best.
Oh, New Orleans was awesome. Yes.
We just made our way across the city.
Cindy was my designated drinker. I mean, like they have designated drivers, right? The person who does not drink so they can drive everyone home. And in New Orleans, you have these bars with really nice music, but they have like this one drink minimum. So I have to drink something. But I didn't drink. So we would go there, she would drink her drink, and she would drink my drink. And then we go to another bar to listen to some more music. So she was my designated drinker.
I'm very good at that.
yeah, you show yourself to be very adept at it. I was really impressed.
And that was before kids, so you know.
Well, yeah.
Now I'd probably be even better.
Well, yeah, you've managed to learn how to deal with sleep deprivation like a pro. I mean, I've went through that myself, so yeah, it's much easier now.
All right. Well, eventually we will meet again.
Yeah.
One of these days.
Yeah. Okay, and then we're gonna meet some of these people. There's some really interesting people as well.
Hmm?
Yeah. And when you were in DC, did you meet any of my students? I think, no, Fernanda was in, no, she was in San Diego. Yeah. So no, we did not meet any of these people, but we're gonna at some point. Okay,
I don't think she understood, Bruno.
I didn't fully catch it.
No, what I was saying is that I was wondering if you have ever met one of my students when we met in United.
Met us.
Yeah, them.
I feel like I've met one of you, but you know, I'm trying to think of the last time you and I were at the same meeting.
It was DC. You were pregnant.
Oh, so that'd be 6 years ago.
Yes, but by then I think none of them were my students. So probably.
No.
Yeah.
Because I know we were with some of your students and some from your collaborators lab.
Yes, yeah, I think you met Kleber, who sometimes comes to these meetings, but not today. Who else? No, it might be just him. Yeah. So.
Yeah, and you know, that meeting too, I got the flu. And so like, not only was I pregnant, but I had the flu. And so I remember just like after the JB Johnston Club, spending most of neurosciences in bed.
Oh, wow, that sucks.
I was really sick. I had to go to the emergency room.
Really? I didn't know that.
Oh, it was so bad. And they said both of my posters were the same day. So I had 8 hours of poster. And literally my boss just had 2 chairs, one for my butt and one for my feet, and had someone, you know, was presenting. I was like, it was
yeah, that's some commitment.
A super glamorous meeting for me. It's a wonder why I didn't go to any for a little while.
Yeah, right.
And now I've been going to SICB, Society for Integrative and Comparative Biology, which I think that's just US, but it's much smaller, you know, because there's the problem with neuroscience is that you're around 40,000 of your best friends.
Yes, that's overwhelming. I remember the first time I went to neuroscience. I mean, my first meeting before, my largest meeting before then was a cosmology meeting. I had 3,000 people. And I thought, that's humongous. It's like enormous. There can't be that many people in any scientific subject ever. And then go to neuroscience, like 30,000. It's really overwhelming.
It's massive, and I hit a point where it's hard to go, and it's hard to take students, and it's expensive, and I was like, I'm gonna try other ones.
Yeah.
So I've been doing like little different subsets here and there, like just trying to find like what is the best. But SICB is really good, and Society Experimental Biology is really fun too. That's where the physiology nerds hang out. They would love zombies.
Oh really? Well.
Oh yeah.
Might go there and present something for them.
It's in Prague. The next one is. So.
Well, let me know. I mean, I'm gonna forget it in about 5 minutes. But I mean, when time comes around, remind me again. I might actually go. I was supposed to go to this meeting in Sicily in May. I was really looking forward to it about neurodevelopment because I'm actually doing something like that now, and proliferation pathways and the like. But of course, it got canceled. And then next year, there's this meeting in Spain, the northern Spain. They have this archaeological dig right next to the meeting venue. And we get to visit that. And it's about glia cells. Yeah.
I was planning on traveling a ton this year, and
yeah, that's really frustrating.
You know, it happens, and to be honest, like
well, it happens every 500 years. I mean, it's not like pandemics don't just happen. Even getting a flu happens, right? I mean,
it's leading to its own different venues for research. So I'm okay with that. Okay, I'm not okay with it, but you know.
Yeah, and you have to learn to live with it. Okay.
Yeah.
Right, people. So I think that's a wrap.
All right.
Yes.
Cindy, thank you so much. Bye-bye, gente. Até quinta que vem.
It's wonderful to see you guys, and hopefully someday we'll meet in person.
Yeah.
But I can say I get to talk in Brazil, and that like makes me really
Okay, well, welcome to Brazil.
Right. I didn't even go outside. I was just at the meeting all day.
Well, that's a really good thing to say. I mean, you went to Brazil and didn't even step outside your room. But that's how much dedicated
exactly
to science.
That deserves like a tattoo or something.
I went to Brazil and all I got was this lousy, I don't know, not even a t-shirt. Thank you email.
All I got was this lousy Zoom recording.
Yeah, okay, that will work.
All right. Well, I will thank you again for having me and listening to me ramble.
Okay, bye-bye.
See you.